Perfect. Good morning, good afternoon, good evening. Thanks for joining us. This is Manoj Dadlani from Cosmos ID. I'm here with Dr. Raja Sivamani, and we're going to be talking about the gut-skin axis in the microbiome field. I'll just kick off by just giving a quick introduction. All right. So this is piggybacking off of our Unlocking the Skin Microbiome webinar, which was on the end of March of this year. If you'd like to see that, you can view it from our website. So feel free to go and see the recording of that webinar. So I'm excited to have Dr. Sivamani in today. He's got an amazing background. Honestly, I don't know how he does as many things that he does on a regular basis. So he's a board certified dermatologist and an Ayurvedic practitioner. So really unique profile right off the bat. He hosts a wellness platform called Jivom. He's does medical and surgical dermatology at the Pacific Skin Institute. He also does integrative research in aesthetics in his clinic, Zen Dermatology. And then on top of that, he's got academic affiliations and research affiliations, doing clinical research at the Department of Dermatology at UC Davis, as well as being the co-director of the Microbiome Research Initiative at the California State University at Sacramento. So wealth of microbiome experience, wealth of skin and gut experience, and looking forward to his presentation shortly. Just before we dive into that, I just give you guys a quick overview on Cosmos ID. So as some of you might know, Cosmos ID has two legs to it. One is we're a full service NGS laboratory that's CLIA certified. And we work in metagenomics, metatranscriptomics, doing things like 16S sequencing, shotgun sequencing, metatranscriptomic sequencing, short reads, long reads, with really optimized, standardized wet lab protocols. And then the other side of it is really our bioinformatics side, which is kind of the core of Cosmos ID, where we've built the largest curated databases for bacteria, virus, fungi, protists, as well as functional characterizations such as antibiotic resistance and virulence factors. And what we're known for is being able to very accurately identify down to the strain level. So we work with companies, with researchers, end to end. So we're involved in study design, helping with sample collection, doing sequencing, bioinformatics analysis, as well as interpretation and custom reporting. And that covers microbiome profiling, strain subtyping, infectious disease diagnostics, as well as applied testing. And again, really the core is, well, once you generate this great amount of big data, how do you analyze it and interpret it? And that's really kind of the foundation of where Cosmos ID started as a company when Dr. Rita Caldwell founded it in 2008. So just diving into that part a little bit, we've done, again, focusing on being able to get down to the strain in terms of resolution. We've built the largest curated databases for all types of microorganisms. And what's unique to this is one is that we've curated these databases. So we've really gone and dived into how taxonomy is defined, adapter sequences that might be contaminating genome sequences, host reads that might be in genome sequences that might be giving you wrong calls, and really diving in genome by genome and curating the data that goes into our database. And then the second part of it is we've organized it as a really large phylogenetic tree, which allows us to have biomarkers that span that entire evolutionary hierarchy to distinguish between different organisms and really get us that higher level of resolution. And then the last part of it is to improve the algorithms we've built. We've done a lot of machine learning on tens of thousands of various samples to reduce false positives and false negatives. So ultimately giving us very accurate strain level resolution. When you look at kind of results, this is some internal benchmark data based on a paper that came out of Cornell, and common classifiers that are used in the field. At the species level, we outperform all of those classifiers when we're looking at a combination of precision and recall, or similar to sensitivity and selectivity. But where you really see where Cosmos ID shines is when we get into the subspecies or strain level. You can see that we maintain a very strong level of accuracy, while tools that perform well at the species level start to drop off on their performance at the subspecies level. Again, this is internal benchmarking. So we took those data sets from that paper and ran our analysis on them. But in terms of third-party validation, we've also entered multiple third-party blinded challenges, and we've actually won all of them. So we entered the Janssen Pharmaceutical Strain Analysis Challenge, and we placed first place on that one. In the Precision FDA CIFSAN Pathogen Detection Challenge, we had the top score in strain level accuracy with the lowest false positives. And in the most recent Precision FDA Challenge, the CDRH Biothread Challenge, we actually ranked number 1, number 2, and number 3 overall against all the participants in that challenge. So really good third-party validation in terms of the accuracy of our bioinformatics. And then what we've done is we've made it really easy for people to go in and analyze samples. So we have a software layer and a visualization layer that sits on top of the platform and allows you to dive into samples one by one, but then also compare samples by cohorts. And I'll do a really quick demo of that and then hand it over to Dr. Sivamani. Let me just switch screens here. And we're live. All right. So I just put up a really quick demo here of just comparing some different body sites to each other. So a stool sample, a vaginal sample, and a tongue sample, and how you can really look at comparing samples on the fly. And we joke around and we say that we try to make bioinformatics at this first level easy enough for our accountant to be able to do it. And it's really just as easy as pointing and clicking. So you can have a heat map view. In the comparative analysis, you can switch between different levels of taxonomy. You can go all the way down to the strain level. You can look at stacked bar graphs and compare samples to each other. So you can see in this case where we have these are stool samples, they're quite different than vaginal samples, which are quite different than oral samples. Then you can also aggregate them. So if you want to say, okay, well, I have this cohort of stool samples, this is kind of what they look like overall. This cohort of vaginal samples, this is what they look like overall. And this tongue sample, this oral sample, is what they look like overall. And what's interesting is you can obviously see that they're very different, but there's also organisms that have commonality between these different body sites. And I think that's really what we're going to start getting into in the next series of webinars, is trying to understand the interplay between different body sites. So today we'll talk about gut and skin. And from an overall profile standpoint, if you're looking at, let's say we look at a beta diversity, you can see that these blue organisms, these blue profiles, which are stool, are quite different than these green profiles, which are vaginal, and quite different from these purple profiles, which are oral or tongue samples. Again, you can look at it from a Jacquard or Bray Curtis standpoint. So that's just a quick intro on Cosmos ID. And I'm going to hand it over to Dr. Sivamani to talk about the gut-skin axis.
Thank you so much, Manoj, and pleasure to be here. I love what you guys are doing with the webinar series, so I'm happy to be a part of it as well. Let me go ahead and get my presentation up and loaded. You can see that okay? Hopefully that, okay, great. So what I'd like to talk about today is we're not going to cover the entire gut-skin axis, but I'd like to hit a few key points. And really the objectives for what I want to do is just build this whole idea of the mechanistic pipeline between the gut and the skin. What is the communication pathways that are in there and how are they communicating? And I'll go through a couple examples for that I think are really salient. And I want to focus on diseases where we use a tremendous amount of antibiotics. And I want to talk about why this whole research is much beyond just technicalities or curiosity. It drives patient care, but also drives public health issues. So I want to point those out, and I'll talk about some clinical evidence for interventions that could be helpful as we go along. So to start it all off, and many of you may have been familiar with this case, but I think it's a really interesting case that just tells us that there's a lot more that we need to understand. So this was a patient, long-standing alopecia areata, which is an alopecia universalis. They're autoimmune conditions where you lose hair in patches And when you have alopecia universalis, you lose the hair over the entire body. And in that kind of a situation, what happens is that you try to do different approaches to regrowth, immunosuppression, whatnot. This patient here, and there were two patients that were reported, they tried a lot of different things. They tried immunosuppression. They got to the point where the patient said, okay, you know, I've come to just deal with this as being a part of who I'm going to be. I'm not going to grow my hair back except for this kind of patchiness that's there. And they moved on, but they ended up getting these bad infections for some other unrelated reason. And they had C. diff infections, didn't respond to antibiotics. They were given a fecal microbiota transplant. And this was like 10 years after they had already resigned to the fact that their hair wasn't going to grow. And what happened was their hair grew back. And so then the question is, what happened there? Is there some constant education that's occurring at the gut skin axis, or did the microbiome shift and the community now shift that related to autoimmunity in some different way that had been constantly in a negative fashion that now has shifted into a different format? So a lot of questions, not as many answers, but very exciting. So the other reason I think skin is very important is that 50% of doctor's visits involve a skin condition. This poor baby has eczema, but if you look at acne, that's like 90% of the younger population. So when we start thinking about the microbiome, we know that it's definitely housed very deeply into many different sites on the body, not just the gut, but the gut is huge. The skin is huge too, but when we talk about the gut-skin connection, we're not just talking about what's in the gut. What we're trying to understand is how is the gut communicating beyond the gut, and how is that communication signal getting to the skin? And so when we think about the different mechanisms that may be in play, we definitely have to think about the immune system. That's very important because the immune system can incorporate things like Tregs, T17 cells, things that are going to control and be checkpoints to what can happen distally from the gut. And the gut is so rich in these sorts of communication instances. But then you also have direct absorption. You take something, you directly absorb. This, by the way, used to be the classic way that we used to think about how the gut might communicate to the rest of the body. You had to take things in at sufficient quantities that would then get absorbed. And a lot of the complaints or the concerns with previous studies used to be, oh, you weren't taking this in a high enough dose, so it can't have an impact. Now we know that's not how it works, because you can actually modulate the gut microbiome even if you don't absorb, and that can have an impact. So whether you're absorbing or indirectly secreting some mediator, there's a lot of modulation of hormones, some classic ones, IGF-1, insulin, those are the classic ones. But you also can think about, other hormones that may be involved, or any sort of intermediary signaling. And that leads to this point of what are the novel signals that may be released from the gut that might be then contributing to this milieu of hormones or signaling factors. So whether they be short chain fatty acids or long chain saturated fatty acids, lipopolysaccharides are talked about a lot. And then now we have this whole notion of testing to see if you have, quote, leaky gut, by looking for antibodies against lipopolysaccharides or different sorts of antibody mediators. We'll spend a bit of time looking at the signaling factors much more than the actual gut microbiome, because I want to shift the conversation away from just looking at the microbiome, because our question always when we see something in the gut isn't what's there, but what's it doing. And so we want to move that conversation in that direction. Now, before we start talking about that, one of the questions that we discuss is, can the bloodstream communicate to the skin, or is it all indirect communication? And the reason this comes up is when you look at the skin anatomy, it is placed topographically with an epidermis superficially and a dermis as you go down deeper. And the epidermis is largely devoid of any sort of vascularity. And that's because it turns over frequently. However, when you go down into the dermis, it's very rich in vascularity, and you have several appendages that do communicate with the bloodstream. So you have the sebaceous glands, which are up here right next to the hair follicle, and you have the sweat glands. And there's two kinds of sweat glands. There's the apocrine and the eccrine sweat glands. And all of them communicate with the bloodstream. And we know this because, one, they have the sebaceous glands are modulated by hormones, which are largely spread through the bloodstream. But we also know that when you give people certain medications, for example, a classic one is fluconazole. When you give fluconazole, which is an antifungal to a patient, and you're telling them, okay, we want to use this to treat certain sort of antifungal issues on the skin, what we do is we give them the prescription, and 30 minutes later, we tell them 30 minutes after you've taken your dose, go exercise or do something that'll make you sweat, because we know it's delivered through the sweat gland. So we know there's definitely a communication. So we know the gut communicates to the bloodstream, and the bloodstream can definitely communicate to these appendages, and the appendages then can communicate up into the skin surface, and not only that, down the hair follicle as well. When we think about the gut-skin axis, I alluded to the immune system. I just wanted to show you a diagram, not to go through it because it's a little bit busy, but the idea being that you have Th17 cells. What are the kind of conditions that are involved in Th17? Well, psoriasis is definitely one of them. Another condition that we've been looking at much more now is hidradenitis suppurativa. There are a few clinical trials looking at inhibiting overactivity of the Th17 cells. But you also have the Tregs, which are important in controlling autoimmunity and also keeping the immune system in check. And so there are many factors that can come in and interplay with this, such as the short chain fatty acids, or if it's some sort of a commensal bacteria that's going to influence. But the idea being that you have a local immune system that is very rich and can speak to the rest of the body. But let's focus in on some diseases, acne and rosacea being the real classic ones. And the reason we are looking at that is because antibiotics are so commonly used. And the first question that's asked is, why are antibiotics used? They are good. There are good studies around antibiotics. They're good at actually having an effect. They're very efficacious, but they're also bad in the sense that if you're using doxycycline, minocycline, which are the classic ones, you can not only build up resistance for the bacteria, say in acne, and acne is not an infection, it's a colonization, but that C. acnes, which used to previously be called Propionibacterium, but now it's Cutibacterium, they can develop resistance to the antibiotics, but probably much more importantly is you can have issues with the bacteria in your gut shifting, and you can also develop drug-resistant bacteria that are pathogenic, like methicillin-resistant Staph aureus. One of the ways we treat it is doxycycline minocycline, and we see that as you use it more and more, it becomes resistant to doxycycline minocycline. So we want to try to minimize antibiotic use. I think many of you are familiar with this. So let's talk about acne really briefly. So what causes acne? Well, you have an overabundant production of sebaceous secretions, which are sebum. And then you can have accumulation within what's known as the comedone. So you get this plugging that occurs right at the hair follicle. And so you have a buildup of the sebum, and you have an overcolonization of the natural bacteria that are in there. Which bacteria involved, we're not sure. But one really important point right out of the gate I want to just mention is most of our sampling is surface swabbing. You can see here that surface swabbing may not even be relevant to the acne environment, except to tell you what kind of species may be present. But when you're really looking at acne, you need to have follicular sampling. You have to, because the folliculars, the environment there is going to have less oxygen. It's going to be much more anaerobic when it gets clogged off as a pore. And so when we're doing surface swabs and making decisions on surface swabs, just realize that there is a major limitation when you're not considering what's going down the hair follicles. And then of course you get inflammation. So the key factors in here, as you can see here, just, so that we outline what are the different factors in here. Obviously, a lot of what we're looking at when we're looking at microbiome is what's the colonizing factors. Is it really Cutibacterium? It turns out when you have other bacteria like Staph epidermidis, which are considered to be anti-inflammatory, or at least they help control inflammation, when you put them in an anaerobic environment, they can actually produce quite a bit of inflammatory factors. So it's still not totally clear cut which bacteria are the ones that are in the etiology of acne, although there is, you'll see a lot of strain type studies talking about Cutibacterium. Okay, so if we're going to talk about gut-skin axis, I do have to mention nutrition as a starting point, and then I'm going to build in some of the factors that tie into the microbiome. So for starters, and this was the second mechanism that we talked about, was direct absorption. How do the foods that we eat and the hormones that they influence affect our skin? So let's start by looking at acne. And the classic ones are milk peptides, oligosaccharides. So when we look at milk, and milk is the classic one that we've really focused in on from epidemiological studies. This was a very interesting study where they looked at people and they followed them as they consumed milk. And a lot of the milk that they consumed was predominantly whey protein. It was done in Brazil. And what they did was they followed men and women over 2 months as they consumed milk as they want to. And they looked at the total lesion counts and they wanted to track what happens to their lesions. There have been many epidemiological studies that have looked at in reverse and said that skim milk is potentially related, or it could be whole milk, or it could be cheese. So I'm going to show you a meta-analysis that's looked at that. But look at the amount of counts of acne lesions as they go up. So acne is counted as total lesions. Typically, you have non-inflammatory and inflammatory lesions. you're starting with someone that has acne at baseline. So remember, milk isn't going to magically cause acne to appear in someone that doesn't ever have an issue with acne to start with. But in those people that had acne as something that they dealt with, then 17 lesions is there at that mild to maybe even moderate, depending on how accentuated those lesions are. You're getting almost a tripling of those lesions as you go over 2 months. And then whether they're male or female, you had the same sort of an impact anyway. And this was a meta-analysis that looked across over 75,000 children, adolescents, young adults, and they looked at what are the different features of dairy that are associated with acne flares and or worsening of acne. And you can see here to the right means that there's a higher association, higher odds with acne worsening. And unfortunately, across the board, it's all dairy in this meta-analysis. Now, you always take meta-analysis with a grain of salt because these are more either patient recall or some sort of epidemiological approach. It's not going to totally nail down a causation. But that being said, it's highly associated, and you can see there are multiple associations across the board here, whether it's all dairy, full dairy, whole milk, low fat. The reason skim milk becomes an issue much more is in many states like California, where I'm located, when they have to pull the fat out, they put in a filler into the skim milk, which can actually be enhanced in whey proteins and oligosaccharides and other factors that may actually stimulate this acne pathway a bit more. Whey protein has been something that's been of particular interest because it does spike the insulin pathway much more. It's part of the reason why you take whey and you build muscle. So these were case reports, and you're never going to find a prospective study on whey. I don't imagine one IRB that's going to allow this at this point, because the whole premise would be it's going to worsen acne. The only way you can do this is association studies or perhaps some follow studies where you're looking to see if there's an association with worsening of acne, like in the previous study that I showed. But there's not going to be a prospective study. So anyone that says there needs to be prospective studies, you're kidding yourself. They're not going to happen. No IRB is going to prove it. But that being said, this was a case where they had a group of 6 males, and they were flared. They stopped them on the whey protein. Their acne got better. And then the physician said, well, let's see if we put them back on the whey protein, what happens? They had another flare, stopped them on the whey protein, and it went down. So this is one of the clinical pearls that we talk about is when we talk to patients that are coming in with acne, especially if they're men, we ask them, and women too, because a lot of people are working about, especially men that are trying to put on muscle. It's not that they're taking steroids. They may be on whey protein. And so we actually have this as a common conversation topic. We know that the high glycemic diet is involved as well. Again, that insulin pathway, IGF pathway is involved. And we know that high glycemic diet is related to worsening acne. And in fact, they've done studies now looking at low glycemic load diets that could be potentially helpful for acne as well. So we've modulated this whole system of this gut-skin axis from a food perspective. We know that Western diet, which is called the standard American diet, which is also known as the SAD diet, is going to drive some of these hormonal factors. And one of the hypotheses is that it'll be driven through mTOR, the mTOR pathway, mTOR1 pathway. And there's some question about whether mTOR2 is involved too. But you can see if there's high meat intake, that's something that hasn't been studied extensively for acne, but probably perhaps is worth studying. But high milk and dairy intake, high glycemic load, these can all tether in through the mTOR pathway, in addition to just having that insulin driving factor. But the idea being that it's going down, and eventually SREBP is one of those transcription factors that's involved in lipid synthesis. I'm sure there's more to be discovered here, but this is kind of a unifying approach that we're starting to think about. So if you're thinking about what are some ways to address the gut-skin axis, are you going to think about foods that may be inhibiting this pathway, help controlling this pathway, or selecting foods that don't drive this pathway as much? But let's talk about the gut microbiome and some of the intermediary factors that may be involved in acne too, since that's part of the reason that we're all interested in this. So this was a study looking at gut microbiome changes in folks with acne. They had 31 participants with moderate to severe acne. So now you're talking about acne that's a little worse, not mild, because mild can fluctuate back and forth, but moderate to severe is pretty fixated acne. Now, you're really not going to have any disease-free days. That's just not part of the vocabulary without some sort of an intervention, whether it be nutritional, but typically you need some sort of pharmaceutical intervention. They also had 31 matched controls that were age matched as well. And so what they found was there were a few bacteria that started to emerge as being much more prevalent in the acne group. And they were deficient in the acne group, I should say, rather. So you had bifidobacterium and lactobacillus, which we know are important for producing certain short-chain fatty acids. Butyrococcus is going to be one that's really important in producing butyrate. We know that's important for gut health. I'm going to talk about the short chain fatty acids. We're going to come back to that because there's more than just butyrate that you need to think about when you're looking at the gut-skin axis. And so what we're starting to see is now we're starting to see that who's there question is there's a slight shift. And we know that there are some shifts. What does that mean? And what was interesting in the study is they pointed a couple out that actually have been tried as interventions previously and shown that there was some deficiency of those bacteria. Now, these are genus level bacteria, and we all know that you have to get down to the strain level, but at least this is a first starting point. And then what we need to do is start looking at what strains are differentiating and what are those strains doing. But to keep moving along in terms of the gut-skin axis, to come back to what I alluded to in the very beginning when we talk about indirect mediators, you think about LPS coming from the gut. When we look at what it does on the skin, it activates TLR4 receptors, which we know can stimulate inflammation on the sebocytes. That hasn't been shown bona fide yet, but I want to build up some of the motivation for the studies that I'm going to talk about next. You have long-chain saturated fatty acids, high-fat foods. If you're eating foods that are basically like junk foods, they're going to be loaded with long-chain saturated fatty acids. They're TLR2 receptor agonists, which we know is already a known pathway for stimulating inflammation, especially on the sebaceous glands. In fact, Cutibacterium acne stimulates TLR2 receptors. So that's already a well-defined pathway. And then you look at short-chain fatty acids, butyrate, propionate, acetate. There are many transporters and receptors. The effect on the sebocytes was largely unclear. We've done some studies. I'm going to show you some partial results. But we're starting to understand that these short-chain fatty acids are important to know, not just for gut health, but they could actually have an impact elsewhere as well. So I want to talk a little bit more about those short-chain fatty acids. So we have acetate, propionate, butyrate. We know they get in through the gut. They're released by the microbiota, and then they can have effects on distant organs. This is a great schematic. Notably, skin is missing, and we're hoping to add to that conversation, but you can see that there are potential impacts across the entire body. When you look at acetate, butyrate, and propionate, there's a couple of things to keep in mind. So butyrate is largely used up in the gut and the gut lumen, and it doesn't get too much into the portal vein because a lot of it is used by those enterocytes. When you get to propionate, a little bit different story. You are getting some that's getting out to the passed into the portal vein and into the liver. Some of it's probably even getting synthesized at the liver level. But when you look at the short-chain fatty acids that are circulating at the systemic level, especially when you're looking at the venous level and in the bloodstream, it's actually acetate that's actually much more out there in the systemic levels than butyrate. And I'll show you some of the studies that we've done, even in acne, and the levels of acetate are much higher. So do we need to start thinking about acetate when thinking about gut-body or gut-skin axis? Everyone is so focused on butyrate, which is obviously important, but should we be looking at other mediators that may actually have an influence elsewhere? So when we start looking at what are the influences of long-chain fatty acids, when you look at sebocyte lipid content, these were in cultured sebocyte cell lines. You look at how do they influence the lipid content within the cell. You can see that when you look at control, there's quite a stimulatory factor. Either they're absorbing the lipids and then incorporating them into their interior, or they're stimulating their the pathways to upregulate lipid synthesis. It turns out, I'm not showing the data here in the interest of time, but they do stimulate some of these lipid synthesis pathways as well. You can't exclude the fact that they may be absorbing the long-chain fatty acids and holding them in the cells as well. Now, if you look at the short-chain fatty acids, it's a much different conversation because, again, you're looking at 8-fold differences here. But when you're looking at short-chain fatty acids, acetate, almost no change, butyrate, maybe a slight bump, and then propionate, almost no change. And if you look at COX-2 and the LOX pathways, they're stimulatory. The long-chain saturated fatty acids are much more stimulatory of those inflammatory pathways. And what we found with the short chain is actually you have a decrease in the synthesis of the COX-2 enzymes and the 5-LOX enzymes. And so perhaps they actually have an anti-inflammatory role. Here's where it gets very interesting. So we've done a series of studies where we've looked at acne and people that have acne and compared them to age-matched controls. When we look at long-chain saturated fatty acids, maybe they're all eating the same bad diet across the board, but we didn't see any shift in the long-chain saturated fatty acids in a variety of the species for long-chain fatty acids. I'm showing C14, C16, C18 here. Now, when you get to the short-chain fatty acids, two things to keep in mind. first of all, look at the levels of propionate and butyrate. If you look at this y-axis, it is magnified in from 0 to 2 for propionate and butyrate, just so that we can show that. But then we have a break, and you're talking about almost a 50-fold difference in what the levels of acetate are in the serum. And what we find is that there is a deficiency in those with acne by about almost 33%. Now, we actually did another study and followed up on this, just comparing moderate to severe acne. These were not mild, moderate to severe acne versus those that didn't have acne. And what we found was you have a deficiency in acetate. So then one of the natural questions that's going to come out of this is, we give people doxycycline so often as a treatment. Is that impacting the short chain fatty acids that are in the system? Are we actually influencing the gut microbacteria in a way that may actually be beneficial by giving doxycycline? Because one thing that to keep in mind is that not all antibiotics are created equal. Some are going to clean out the anaerobic bacteria and some are not. Doxycycline does not have good coverage of anaerobic bacteria. So it's probably not going to have as profound of an impact on the gut. But what we wanted to look at was what happens in at the short chain fatty acid level. Now, there are studies showing that doxycycline, when you take it, you still do impact the gut, and that impact can be up to 6 months, and some people even longer. So I don't want to imply that there's no impact on the gut. There definitely is, but it's not as profound as, say, if you take clindamycin, which is where you have the big risk for something like C. diff. Okay, so what we found was, just to remind you, there was a deficiency in the acetate. The people that were on doxycycline, their acne definitely improved. We already know it's actually an indication for using doxycycline is acne. What we found is that there was a bump. There was an improvement in the plasma acetate concentrations. Sorry, I had serum in the previous slide. It's actually plasma that needs to be corrected, but it is plasma concentrations. So plasma acetate concentrations bumped a little bit, but always be wary when you zoom in on the y-axis. Here you can see, although there was a trend, it was a very tiny bump. So doxycycline doesn't seem to be shifting the short chain fatty acids that much, or at least acetate that much, even though it had an impact. And so we think maybe doxycycline has more of a direct anti-inflammatory effect, which has been shown in other studies, but doesn't seem to be influencing the short chain fatty acids as much. Well, what about the role of probiotics? Well, we did another study where we looked at what happens to acne when you give them probiotics. And the probiotics that we used in this particular study were spore-based probiotics. And it was 5 spore-based probiotics that were put together, and they were supplemented. And what we found was, again, if you look at the black bar, this is overall acetate levels. The people that were in the acne group, this was a sub-analysis in a bigger group. And so what we did was we only found a trend here, but it recapitulated what we found previously, which is that the acetate levels are deficient in this particular case. And what we found was that when you gave people probiotics versus the placebo, you were able to replenish the acetate levels in a trend here. But when we looked at the acetate propionate levels, you were able to restore levels back towards normal much more than what we saw with doxycycline. And the key question is, did that impact the acne? And it did. Because when you look at baseline and placebo, no shift in the total lesion counts or the inflammatory lesions or non-inflammatory lesions. However, when you looked at the probiotic supplementation group, which was only over 4 weeks, you started to see there was a decrease in the total lesions, decrease in the inflammatory lesions, and the non-inflammatory lesions. So our finding there is not something that's only the first time it's been reported. Actually, back in 2013, there was another group in Canada that looked at 3 groups, probiotic, minocycline, and the combined group. And they looked at what happens when you give people a combination of lactobacillus, bacilli, and bifidobacteria. And if you remember, in that gut microbiome study, 2 of the bacteria that were deficient compared to the controls were lactobacilli and bifidobacteria. So now they're going into actually giving this as a supplement. Here's what the data showed. If you look at, just to walk across here, blue is probiotic only, red is minocycline, which is antibiotic only, and green is the combined group. When you got to week 4, similar to what we found in our study, you started to see an improvement in the probiotic group, which is the blue group. What was really interesting is that it was keeping pace with the minocycline only group. And this was in women that were aged 18 to 45. So this was more adult women. And the combined group also seemed to have the same sort of effect. they carried this out to 12 weeks, which is a very well done study, because normally when you look at all of the endpoints in acne, you have to take them out to 12 weeks. By 12 weeks, you saw that the probiotic only group did as well as the minocycline group, and the combined group actually synergistically was doing better in total lesion counts. If you look at inflammatory lesions, same sort of a story that you saw here. You see a drop, and you see that there's an enhanced synergistic effect when you have the probiotic and minocycline group together. And finally, for the non-inflamed lesion count group, same sort of a deal. And here, maybe the non-inflamed lesions wasn't as synergistic, but the idea is that the probiotic group was keeping pace with the minocycline group. Here's what was very interesting. The group that got minocycline, a portion of those women, and we know this in clinical practice happens too, got vaginal yeast infections. In the probiotic group and in the combined group, there were no vaginal yeast infections that were noted. Now, the study wasn't powered to look for vaginal yeast infections. It was more of a side effect that was being reported. But I think it's notable that there were 0 women in either of the probiotic or the combined groups that had any vaginal yeast issues or basically vaginal candidiasis. How is the gut communicating to the vagina is something that will be very interesting as we start to uncover these sort of indirect influences. But it's a big deal, because a lot of our women that we do put on antibiotics do talk about this. And now the guidelines in acne are to get off antibiotics. So I think it's interesting to see how probiotics may come and fill in that landscape. So that's a little bit of a background with acne, just to show you that there are now probiotics that are on the market that you could utilize. I'm just not mentioning them here, but these are some of these are commercially available. If ever anyone wants to know, I'm happy to answer that later on. Now, rosacea is another condition that's not quite like acne, but slightly different. However, very inflammatory. You can see here in this image, there are many different variations of rosacea, but you can have some very inflammatory variants where a lot of pustules, papules, crusted lesions, and redness and swelling. And it tends to occur in folks that are in their 30s, 40s, 50s. And there are a few causes here. One is innate immunity being in overdrive. Another one is ultraviolet radiation. You also have vascular reactivity. The inflammation is in overdrive. The epidermal barrier is now in dysfunction. It's a chicken or the egg. Did the inflammation lead to the epidermal barrier being working in a dysfunctional status, or is it the other way around? And what is the role of the local microbes like Demodex? And actually the Demodex mites have their own microbiome. And is that bacillus that's in there and within that Demodex, is that influencing it? So these are all the thoughts that are around what might cause rosacea. And what's now been added to this conversation, which I'm going to show you, is are there altered states in the gut microbiome that may be influencing rosacea as well. So here are some of the patient-reported history of foods that trigger rosacea. And I think it's always telling to start by talking about nutrition, because again, gut-skin axis, nutrition is an important factor. There hasn't been prospective studies or epidemiological studies at the level like they've been done in acne, where we've started to tease apart maybe things like dairy could be involved or high glycemic foods. Here, it's more about patient reports, but things that stand out, things that are very acidic or they're very spicy, they tend to trigger rosacea. And that may not be a gut skin thing. That may be just through the TRPV channels that are locally present. We know that high temperature, if there's coffee or if it's tea, it's not the caffeine itself, but it's the temperature of the drink can trigger rosacea. Another one is if you're having spicy foods, that can also trigger rosacea. But there are also other factors in here. If they're acidic, if they're very acidic, like things like vinegar, yogurt, sour cream, they may be involved. And then this, again, these are patient reports, but nightshades, there's a question of whether they're involved. Need more prospective studies, but I want you to see that this is an area that's growing, and this was reported by the National Rosacea Foundation. And this is the schematic just to try to put together all those, what we might think are varied inputs, but they're coming in through activating the TRVP channels. And so, you know, you're looking at things like peppers, whether it's like hot drinks or if there's sunlight, they all can be influences on this vascular reactivity and perhaps inflammation. There have been a few studies correlating dietary factors. The only correlations that we see, and just to walk you through these three on the top here, this is ETR means erythematotelangiectatic rosacea, more vascular reactivity, papulopustular rosacea, meaning you have those red bumps and pustules, and phymatous rosacea, where you have overgrowth of the sebaceous glands. We've unified them as rosacea. These three may be overlapped in any one given patient. You don't have to have them as being exclusive, but we don't understand why someone might get Phymatous rosacea over someone getting papulopustular over erythematolangectatic. Totally, we're starting to understand some of this maybe with vascular reactivity, or perhaps there's something in the hair follicles that's driving it, or something with the sebaceous gland biology. We're still uncovering that. What's interesting is if you have fatty foods that you eat more than 3 times per week, that gives you a higher odds ratio of having erythematolangectatic rosacea or Phymatous rosacea. And that's interesting with Phymatous rosacea because you have almost a greater than five-fold increased odds. And of course, if you're talking about sebaceous glands becoming hypertrophic, are you driving that with the fatty foods? Don't know. Again, these are correlations, but very interesting. A little bit opposite finding here with dairy. With dairy, you see that there's less of a correlation with rosacea, which is very interesting. And could it be that dairy may be bolstering the skin barrier or whatnot? We can hypothesize, speculate, conjecture. We don't know, but it's an interesting uncovering that dairy may actually be protective in cases of rosacea. Again, one study, let's see what comes out as more studies come out. Now, if we want to talk about the gut microbiome, one of the areas that's really been focused in on is the small intestines. And so small intestinal bacterial overgrowth, also known as SIBO, and rosacea is an area that's become of more intense study. And patients with rosacea have been found to have a significantly higher prevalence of SIBO. And usually when you're looking at SIBO, the classic way to do that is to get down there with an endoscopy, take a sample and culture that. That's not practical. And so what people have relied on are breath tests. And breath tests can look at either hydrogen production or methane production, or hydrogen sulfide is the new one that they're looking at. There's been some reasonable sensitivity, specificity. It's not perfect. But what they've also found is that if you can resolve the SIBO with antibiotic therapy, you can have prolonged remission. We're not talking about, because a lot of the topical treatments that we use for rosacea, you use it as soon as you stop, the rosacea will come back. It's a classic issue with topicals. And so I tell people, it's like brushing your teeth. You got to do it every day. If you don't, then the rosacea may come back. You still got to focus in on good sun protection because we know that flares rosacea too. But in these folks, when they were treated with antibiotic therapy, their improvements were maintained, suggesting that maybe there was some imbalance in the small intestines that was used for a prolonged period of time. Now, when you're looking at SIBO and rosacea, there are a couple of factors that are really important. So this was a study that looked at people that had rosacea, and they cleared 20 of the 28 subjects when they were given rifampin and greatly improved in 6 of 28 patients. And excuse me, but you can see that these are pretty severe rosacea, and over time, you can see that they've all cleared. And probably the more important thing is, of those 20 subjects in the placebo group, rosacea remained unchanged. So there was a placebo group and a rifaximin group. And rosacea remained unchanged in 18 subjects and worsened in 2 subjects in the placebo group, whereas the rifaximin group, which I'm showing you up here, 20 of the 28 cleared. But in patients that were in the placebo group that were switched to rifaximin to treat their SIBO, the SIBO was eradicated in 17 of the 20 subjects, and 15 had complete resolution of their rosacea. Now we're suggesting that there is a different factor at play, not just topical reactivity. So rosacea is not just a skin issue at this point. We're seeing that there may be a gut-related influence, and so we have to start taking that into account. So there was another study also that looked at SIBO, and 51% of the subjects with rosacea were found to have SIBO. So again, it doesn't mean that everybody has SIBO, but something that we start to look for, and it's something that I clinically start to look for. And so the people that were given rifaximin here, clear or markedly improved was 45% of the subjects. 25% had moderate improvement, and then 11% had mild improvement. So, all in all, you're looking at almost a greater than 80% improvement by treating their SIBO and addressing SIBO with rifaximin. So could this be the reason that when we give people doxycycline for rosacea, that's helpful because we're also impacting their SIBO, because doxycycline treats SIBO as well. So one of the questions that we need to probably ask is, are there other non-antibiotic approaches to treating SIBO? Can we take that approach either with probiotics or other botanicals or other sort of interventions that need to be coming down the pipeline? We don't have those sort of studies yet, but I think this really highly motivates that sort of an approach. So some general closing thoughts when you're thinking about probiotics. We want to think about how are these probiotics stored, whether they're in the fridge or at room temperature. Do they stay alive with ingestion? Do they need to get past the gastric juices? So if they're encapsulated or encapsulated well so that they don't get killed, and the question is, do your bacteria need to be alive to have an impact? And the studies are showing maybe not. What if they're the spore-forming bacteria? Can that get past the gastric juices better? And the answer seems to be yes. And then they germinate in the potentially the small bowel, or typically a small bowel, but do they get into the colon? And then which bacteria? Can you devise smart approaches? Can you pick something for someone's rosacea versus someone for acne? We don't know, but these are some considerations that we have to think about. So overall, hopefully I've given you a good landscape on the changes in the gut microbiome that are associated with skin health, and that there's several modes of communication. Acne and rosacea seem to have a component that is modulated by the gut microbiome, and there are indirect mediators that we're starting to uncover that can have that communication. It's not just the immune system. The immune system is important, but there seem to be other mediators, whether they are either food or hormones or other indirect mediators like the short-chain fatty acids. Probiotics or targeted antibiotics based on gut dysbiosis seem to be showing promise for the improvement of acne or rosacea, as you can see. And so the probiotic research is still, it's in infancy. We're still understanding who's there and what they're doing, but now we're starting to look at interventions. And I think that this is really the way we need to go, so that we get a better understanding of what bacterial strains and doses we should be using, and so that we can have highly refined bacteria that we're giving to people. And then how do you integrate that with prebiotics? And also one thing we haven't talked about, and a lot of the studies have been largely 16S, what's the functional role of fungi? Are there fungal changes? I didn't talk about it here, but if you have psoriasis and you have heavy growth of psoriasis on the scalp, they've shown that, like SIBO psoriasis, they've shown that those people tend to have a bit more candida overgrowth in their gut as well. And so I think that we're going to start to understand that there is more players in the gut than we originally think, and that they're probably going to be more important in other factors than we had originally appreciated. So we did a review across all different dermatological conditions. If any of you are interested, you can look at how the skin and the gut microbiome are shifted and how they relate to a variety of dermatological conditions. And I do want to just talk about the microbiome research initiative that we have going on. It's myself and Dr. Crawford. We have some funding from some other companies. Microbiome Labs is one of the companies that's been a generous donor, and been one of the main donors, in fact, in helping push forward our research in this area. And the questions we ask are basic science questions. What happens to bacteria? What kind of postbiotics do they make? If we grow them in an anaerobic environment versus an aerobic environment, if we expose them to certain food particles, and how do they influence, like what are the postbiotics that come off, and how do they influence biology around them? So, you know, it's a funny thing Carl Sagan said. He said the cosmos is within us. We're made of star stuff. What he didn't know is that they were just bacteria, and we're even starting to now develop a course for the planetarium where we might talk about the universe within, so that we can start teaching young kids about the microbiome, which really is a universe within ourselves. And so with that, I do want to just thank all of the folks that have helped me put together a lot of the research that we do. I know Manoj talked about the various things that we work on. It's not possible without having different sources of funding, and also all the collaborators that we work with. We have folks that we collaborate in the Department of Nutrition, Food Science, Microbiology. So it's definitely a fun time right now. And as one of my students said, what a time to be alive. He's absolutely correct. And I do also want to just finally plug, if any of you are interested in more and more education around dermatology and integrative approaches, I'm one of the co-chairs, but the Integrative Dermatology Symposium is a virtual symposium, and it's going to be online from October 23rd to the 1st for this year. But it's one where we keep coming back to different sorts of interventions. What are different approaches you can take when you're thinking about dermatology? And they have the early bird that ends tomorrow, in fact. So you might want to get a jump on that. And with that, I just want to, I take some questions. And Manoj, thank you for the opportunity to present in this webinar. I think what you guys are doing is awesome, and happy to add to the conversation. So I know there are some questions. So let's see if I can jump through some of these questions that are here, and we'll go through to the top of the hour. So looks like one of the questions that we have is, what about eczema? Eczema is definitely one that's important when you're thinking about the gut microbiome. I didn't include that in here in this just for sake of time, but we have a systematic review where we look at different sorts of probiotics that have been included. And yes, there are some that are now coming out, like Lactobacillus rhamnosus GG. It has some early results. Bifidobacteria breve has some results. Lactobacillus salivarius, that has some early results. So we're starting to understand that the gut microbiome is probably important. In fact, there was one early study, which I hope there's a follow-up study, where they gave pregnant women Lactobacillus rhamnosus GG as a supplement, and they were high risk for their children to have eczema, meaning either they had eczema or they had previous siblings that already had eczema. And what they did was they tracked these children. They had the women supplement during pregnancy and then supplemented the kids as they after they were born. And by the age of 2 to 3 years, they looked at the people that got the placebo versus a supplement, and it was a 50% reduction in those that ended up developing atopic dermatitis. What's involved mechanistically? That's for us to unpack, but very interesting result. I hope someone follows up on this and we do a repeat study, because you always have to be careful of pilot studies, but very interesting, very intriguing. So the data for the benefits of gut supplements for relieving acne is compelling, but is marked as unpublished. Yes, we've actually got that submitted, and it is now under review. And in fact, we're doing a much bigger study with 60 people right now, placebo-controlled, and that's also underway. So, but the unpublished one is in review. It's been submitted, and so we're hoping that'll get through review pretty quickly here. So in terms of another question here, were there any direct evidence for changes in the skin microbiome or the acne microbiome in response to changes in the gut microbiome, which is probably modulated either by diet or antibiotic treatments? So it's a good question about skin microbiome. Skin microbiome is an interesting one. We didn't do that particular study, but there are other studies looking at, if you give antibiotics, how does that shift the skin microbiome? They're looking at ribotypes of C. acnes. So are there C. acnes, specific strains that are involved in acne much more than others? And they are seeing that there are shifts in those. But this whole gut-skin connection, what we don't have is a clear connection to how, if the sebaceous gland is influenced, how is that influencing the local bacteria? That needs to be the upcoming studies that come out. But largely what has been looked at is just looking at, if you give an antibiotic, what happens. But from a food perspective or nutrition perspective, we haven't seen any of those kind of treatments. Can foods or probiotics reduce skin wrinkles? Well, we just published the first prospective study looking at almonds, and it was in postmenopausal women. And what we did was we supplemented them with 20% of their calories with almonds. None of them gained weight compared to the control diet, which was just like pretzels and basically non-nuts. And what we found was there was about a 9% reduction in their wrinkle formation after 16 weeks. And what we've done now is we've completed a bigger study looking at 6 months, and we'll have those results out soon. And we've done a third study looking at mangoes, because mangoes are so rich in carotenoids. And it turns out that if you supplement with a lower level amount, 85 grams, and this is now in submission, but 85 grams of mangoes every day, you can improve wrinkles on the face. If you supplement with more than that, very high, 250, for some reason, we didn't see that improvement. In fact, there was a slight worsening. So we have to unpack what was going on there. But it's one of those things where we have started to see some foods. Probiotics, we haven't looked at that quite yet. So perhaps they'll be, we've looked at topical probiotics showing some improvements in skin wrinkles, but not systemic probiotics. Have you looked at the effect of milk peptides like beta-7 casomorphin on the gut inflammation and interactions with microbiome? I think that's such a fantastic question. We have to unpack these milk peptides. What we have looked at, and this is a question from Bruce. I don't know if this is Bruce German, but if it is, then I appreciate the question. Hello. If you're looking at milk peptides, we've looked at the beta-casein proteins, and we've looked at the whey proteins and how they influence the sebaceous gland directly in culture. And we see that directly in culture, they upregulate production of sebum, or production of lipids. And they also stimulate the lipid pathway. But in terms of how that influences the gut inflammation, from our standpoint, and how that might influence the skin, we haven't looked at that. But I'm sure people have looked at milk peptides in the gut itself. That's something that so I don't have that information directly on me. It's something that I can easily look up, but we haven't looked at in the context of skin. How good and accurate are we in modifying the gut skin microbiome right now? I think the question there should be, how good are we in accurately portraying what's in the gut right now? And largely the studies have been 16S. And, is that going to give you the full snapshot? Probably not. Have we talked about fungus enough? Probably not. Have we looked at what the virome is and how that's contributing to the overall microbiome? Probably need more there. So I think we've gotten okay, is what I think, at looking at what the gut and microbiome are doing. I think in terms of modifying, though, I think we have a lot of tools that are present. The foods that we eat, we know that some are going to be much more heavy in prebiotics. We also know that there are certain probiotics now. If you look at systematic reviews, you can see that some of them are starting to emerge out of the literature for atopic dermatitis, or if it's acne. Acne doesn't have nearly as many studies, but we're starting to see certain bacteria that may have an influence. And as we go along, we're going to have to start asking the question of what about synbiotics and what about postbiotics? What influence are they going to have? So I think, we're getting there, but we're not quite there yet. Do you think quarantine has shifted the gut microbiome? Well, absolutely it has, probably, but maybe not in a way that you think is beneficial. People that are quarantining now are probably eating less at restaurants. And believe it or not, if you live close to a restaurant that locally sources, you might actually be eating healthier food than all the prepackaged stuff that people are buying now, because a lot of people are buying prepackaged if they're worried about their produce having some, they're doing like hyper washing of their produce. But if you're cooking more at home and you're cooking fresh foods, maybe that's shifted in a good way. But maybe if all you're eating is processed foods at home, then maybe your gut microbiome's actually gotten worse. And a lot of people talk about COVID-19 leading to the COVID-20, which is, weight gain. So I'm not sure that that's necessarily a good thing. I hope people are still getting out there and exercising, but, so I'm sure it's shifted. I don't know if it's good or bad, though. It depends on what their habits are at home. Other than that, let's see. Have there been any crossover studies looking at what happens to skin microbiome by experimentally changing diet, like the Western diet versus the prudent diet? Brilliant question. We have not done food studies that look at shifts in the skin microbiome. A lot of those studies have looked at the gut microbiome, but not the skin. And so for that reason, looks like I have a striped look for my face. The sun is rising, so I look like a little tiger, but that's all good. But yeah, we have not seen those sort of studies. And I think, let alone the crossover studies, we need some prospective studies looking at this as well. But I'd be very curious to see that. But the answer to that is I'm not aware of any crossover studies. What if we treat acne directly by applying lactobacilli on acne lesions on the skin? Would this recover the lesions? This is such a profoundly great question, because this notion of adding probiotics on the skin is something that's used as a marketing edge by so many companies. And I'd want to unpack that for a couple seconds here. If you're going to put probiotics on the skin, 3 questions. Is it dead? Is it alive? And is it an extract? Because if the WHO definition of a probiotic is using a live microorganism to impact some sort of positive change. If you're putting on a bacteria on the skin, it's usually not alive unless they're refrigerated. Now, the second question is, if you put a bacteria on the skin, is it temporary, is it transient, or is it going to have an engraftment? Engraftment is really, really tough if you're a foreign species. If you're not some species that's native to the skin, or you don't survive on sebum that well, you're not going to make it on the skin. And in fact, we've done studies showing that you can diversify the skin by drying it out. So one of the fallacies that a lot of people look at is they look at gut diversity as a good thing. Skin diversity can go either way. If you're very diverse, it could also mean that you're bone dry, and so that you don't have that abundance of the lipophilic bacteria that normally would be there. So just using diversity as an endpoint without looking at what the sebum load is can actually end up leading to detrimental consequences clinically, because you might just be drying the skin out and people could have a lot of irritation. So going back to the lactobacilli conversation, potentially, but what might be better is if you can take the native bacteria that are there, somehow reformat them and deliver them back to the skin, or take that bacteria and deliver it to the skin, even if it's from another source. They may have a higher chance, especially if they're alive, of taking hold and potentially having a longer life over there. Is Candida auris showing up as a player in skin issues? Candida in the gut, albicans at least, seems to be overabundant in people, like I had mentioned, in those small studies that looked at psoriasis. In the skin itself, we know it's an issue in intertriginous areas. So if you have overgrowth of Candida in like the armpits or the groin area or neck folds, that's where it can become an issue. But one of the problems is we don't really see it as a big player when you have a lot of just aerobic. If it's aerobically dominant, we don't see Candida as a big issue. There are no standardized testing methods for skin microbiome. You are 100% correct. There isn't a standardized testing method for the microbiome in general. If you take any sample and you send them to a bunch of companies, you're going to get multiple different results, which is why you have to look at the pipeline very closely and make sure that they actually have a publication record and that they can walk you through how they're doing things. And also the follicular sampling is all over the place. I mean, the sampling in general. If you're going to do surface swabs versus follicular sampling, you're going to get different results. And how you do the sampling matters too. How hard you press, is it wet versus dry? I mean, there are a lot of different factors that are in here. So, you know, I think that's important. And then there's a point about cosmetic products being tested on healthy skin. That's right. You know why? Because the regulatory bodies won't allow you to do it any other way. Because if you need to test anything else beyond a cosmetic endpoint, for a topical, you need an IND many times if you're studying structure function. So just because of someone that runs a lot of clinical studies, you have limitations. So if you're doing your first pass foray to understand how things change, you're going to come at it from a cosmetic approach because that's just the way you can get it done. So overall, I think there are a lot more questions, but I think that we've hit time. And guys, if any of you want to talk or you want to follow up a webinar, you know, let Manoj know, and I'm more than happy to put something together that can extend what we're talking about. But I really appreciate the time, and thanks again, Manoj, for inviting me.
Raja, thank you. That was phenomenal. I've been getting texts as well as a lot of chats about how great your talk was. So again, thank you for the time. Really appreciate it. Thank you to everyone for joining, and we look forward to our next webinar. We'll email you when that's up and running. But Raja, again, thank you so much.
Of course. We'll see you all.
Good to see you. Thank you. Bye, everyone.